Go ahead and get started. Thank you all for coming back fairly promptly. Appreciate that. Hope that folks are online. A great first half of our symposium, and now we're in the home stretch here. So I am pleased to introduce our next speaker, Dr. Jubao Duan. Jubao is the Charles Walgreen Research Chair and professor at the Department of Psychiatry and Behavioral Neurosciences at U Chicago, and director of Stem Cell and Functional Genomics in Psychiatry, as well as the co-director of our Center for Psychiatric Genetics at the Endeavor Health North Shore Hospitals in our Department of Psychiatry and Behavioral Sciences. I was just telling Jubao when I first met with him a couple years ago, one of the things that struck me in that meeting were two things. One, that I probably didn't understand 95% of what Jubao was explaining in terms of his research, so hopefully we'll get a clearer sense of that today, because it's really brilliant and amazing. And that secondarily, if there was anyone in our department who's going to win the Nobel Prize, it's Jubao. So definitely very excited to welcome Jubao to talk to us about psychiatric genetics and functional genomics in schizophrenia.
Okay, all right. It's a great pleasure to be here. Thank Roman for a kind of introduction, giving me a lot of pressure. I have to win the press. Anyhow, my topic here today would be more like okay, all right. So it's really honor to be on the same stage as Dr. Cook, who spoke earlier. As I mentioned to Roma, when I came here as a postdoc more than 20 years ago at the University of Chicago, Dr. Cook was a professor there. I learned a lot of genetics from Dr. Cook, and also Dr. Alan Sanders who sit there, and also Dr. Gedgeman. We introduce in next slide. So today, I'm really honored to on the same stage as Dr. Cook, and also great pleasure to be here to share my research. So before I go more in detail in science, I want to give credit to Dr. Pablo Gedgeman and Dr. Alan Sanders. Dr. Gedgeman established the center in 2003, more than 20 years ago, when we first moved here at North Shore. He retired a few years ago, and since then, Dr. Alan Sanders and I co-direct the center. So initial research focus of the center was to identify, genes that contribute to schizophrenia risk and other neuropsychiatric disorders using linkage and also association approaches. But recently, I kind of add a new research program, which is one mentioned Roman mentioned, is using stem cell and functional genomic approach to try to connect the GWAS finding to the cellular phenotypes that are relevant to the schizophrenia and other neuropsychiatric disorders. This slide, you have seen this slide. The only difference I compare this slide to the slide Dr. Marvin showed is I have 48% here. I don't know where it came from, but so for identical twins, you can see they share the 100% genetic makeup, but only the chance to get the disease is only 48%, near 50%. That means genetic plays a significant role in conferring the sustainability, but also there are other significant players such as epigenomics and also environmental stimuli, environmental risk factors. So at early stage, as I mentioned earlier, when I joined the team at the University of Chicago, at that time, it's a very strong genetic research program. And we try to identify genes, associated with schizophrenia and other NPD. The hypothesis at that time was like, the disease has to be caused by a bunch of genes, called that at that time called oligogenic model. It's like there are a handful of genes, they contribute large effector site to the disease. So we use kind of a powerful approach for identify those kind of disease, oligogenic model disease, use a linkage approach. Basically, you look at the affect the pairs in the family, or parents and proven how they share the genomic region that confer the susceptibility, that called genetic linkage. The resolution usually is very low, and the power is not as high as association study, which I will show you later. So at that time, we had, I think this is one of the largest linkage study for schizophrenia. So basically, we use more than 400 families here. We identify those regions that co-segregate, well, not co-segregate, they are linked or shared by the affected proband in those schizophrenia families. As you can see, this is like a genome-wide plot. You can see different regions. The resolution is very low, as large regions. You don't know which gene even, it's like a megabase pair region. So this is a region on chromosome 8p, including those genes, as you can see here, like PPP3CC and neuroregulin 1 at that time is very popular risk gene for schizophrenia. So this is the genetic linkage, and then, another approach called genetic association study. So basically here, instead of look at the, shared by affected siblings, affected sibling pairs, you look at the, as a population level, you look at use SNP as a marker to compare like between cases and the controls, whether the SNP as a marker, is more frequent in cases or in controls. So you can see here, for example, this is example, you can see if a allele is, case is higher than controls, this is extreme case, in schizophrenia, we never see this kind of case. It's a very minor difference. But this is extreme case, just to show how the association work. So with this, you can do association test to see whether the SNP marker is associated disease in the population if you compare, cases versus control population. Usually at the early stage, the population, the sample is very small, kind of in 1,000. So, the association signal actually kind of stochastic. Sometimes, we identify gene as associated with schizophrenia, but other study use a larger sample or similar sample, they could not replicate that, which is the case for many of the study, including ourselves at that time. As you can see, at least some gene we studied in the lab, including, one called trisomy receptor at 6q, and neuroregulin 1, that's Manning, and the CUMT, Dr. Alan Sanders and I, we have been contribute to the literature at that time, but later on, it kind of turned out, many of the genes, including the ones, we have tested here, cannot be really replicated if you use a much larger sample. So that means, a lot of association identified use small sample for schizophrenia research at the early stage really, is evidence hanging in a balance. It's not like there's no certain evidence. Until, the GWAS stage, you heard about a lot of GWAS, but GWAS, genome-wide association study basically really opened the research avenue for the schizophrenia and other neuropsychiatric disorders, like 2009. This is a trio Nature paper, including one from our group, led by Dr. Gedgeman. You can see, we identify one locus 16p is significantly associated with schizophrenia at that time. Its MHC region contains HRA genes, so it's like immune-related gene clusters in this region. Other locus have also been identified, but a very small number. Only 1 or 2 genome-wide significant loci have been identified through this, effort, like combined effort. This trio of Nature paper. So this is the breakthrough at that time. So what's GWAS? We have talked about genome-wide association a lot today. So basically, similar to association approach I mentioned earlier. So you have, instead of testing one SNP marker at a time, you test them, in the large scale. So basically, you have 1,000 or millions of SNP marker in a chip, and you use hybridization to interrogate those SNP marker, the allele frequency in cases in controls with in large number, usually more than 10,000 or even million control samples, and then you do the association test, genome-wide, and you determine a genome-wide significance threshold. Anything above the threshold in the Manhattan plot here would be, genome-wide significant association. So this is genome-wide association. This is really unbiased approach to interrogate the whole genome for SNP markers or loci that are associated with a disease. So with this approach, this is the threshold basically for determining whether a signal is genome-wide significant or not. Why use this? Because in the genome you have 5 or 6 million, common SNPs usually. So you use this kind of threshold to account for multiple testing burden. Now, so with this genome-wide approach, since 2009, we have a trio of Nature paper, and start early 2011, and to last 10 years from 2011 to 2012, actually you can see the genome-wide significant loci associated with schizophrenia increased from 5 to actually here 287. In 2014, a landmark paper we reported 108 loci. So this is a Manhattan plot just to show you, which loci associated with schizophrenia. This is from the landmark study in Nature 2014. We, as a group called Molecular Genetic Study of Schizophrenia, MGS, contribute substantially to this PGC consortium. As you can see, there are many high-rise buildings here. The highest one, the tallest one you can see is MHC region, actually. This is kind of, there are, as I mentioned earlier, has HRA gene. So it's kind of consistent with long-standing hypothesis of immune hypothesis. When Dr. Marvin mentioned in North Sphere, you have influenza infection, you may have high risk. So that means, in interplay between this genetic locus and the environmental risk factor may play a significant role in conferring disease susceptibility. There are also other, known gene, known, pathway, for example, glutamatergic gene, and also most remarkably is DRD2 actually is one of the genome-wide significant loci. And, there are some novel loci that may represent a new biology, for example, MIR137, which I will talk a little bit more in my next slide. So this is the GWAS, right, in 2014. But if you have more samples, for example, here, instead of a PGC3, WEAV3, we have 76,000 cases versus, 243,000 controls. You have 287 loci. So with more samples, you can see you have significant more loci that associated with, can be identified to be associated with this with disease, with schizophrenia. If you summarize the major neuropsychiatric disorders, the GWAS result you can see here, up to 2014. Yet Roman mentioned there are not much happening in last year, but actually you can see much more actually genome-wide significant loci identified for bipolar and for major depression. Imagine each loci has many genes. You can imagine can calculate how many genes can be maybe involved in this kind of complex disorders. For schizophrenia right now we still have 287. I bet if you have more cases, definitely you have more loci to be identified. Compared to schizophrenia, bipolar, major depression, PTSD, for autism actually, you only have very few GWAS risk loci, as mentioned by Dr. Cook earlier. Probably ASD, the genetic risk spectrum for ASD may be different from schizophrenia and other neuropsychiatric disorders, more towards neurodevelopmental dysfunction aspect. and also there are more de novo and rare high risk, high penetrant risk variant for autism, although they share, a genetic makeup, as you will see from this slide. So this is the genetic correlation between different neuropsychiatric disorders versus bipolar in the recent publication Nature. As you can see here, the schizophrenia, major depression, and PTSD, they are among the the disease that share the most with bipolar GWAS association. ASD is the among the lowest sharing spectrum. So this is may explain clinically you may see they have similar symptoms. They share some common pathway or common gene pathways. So yeah, that genetic pleiotropy can explain those comorbidity, clinical comorbidity, I think. So this is the GWAS, what GWAS told us. Now, in addition to the common variant, identified the GWAS as a major part of the genetic risk spectrum. There's another a very important risk factor called CNV. Dr. Cook already introduced the concept of CNV, which is basically the large chunk of genomic deletion or duplication in our genome. So what I show you here is one of the most common CNV that associated with schizophrenia called 22q11.1 deletion, also called DiGeorge syndrome. Among those 22q deletion carriers, about a third of them, they may develop schizophrenia. So you can see although they are highly penetrant, but still it's not like, you have the deletion, you will have the schizophrenia. So the value for diagnosis, is tremendous, but still you have to consider, other alternatives here. The reason for those uncertainty of for the diagnosis, prediction of the disease, likely because this, each CNV region expand a large, genomic interval. As you can see here, you have like a 20 megabase pair region. They span more than 40 genes. So any one of those or combination of some of the gene may contribute the phenotype, different phenotype. So you have a spectrum there, a spectrum of phenotype there. So a third of them, they develop schizophrenia. Now, also exome, whole exome study identified those rare risk variant in the protein coding region. They change protein function. Likely, most strong. The strongest risk factor in those exome region is those called ultra-rare coding protein truncating variant, which means they cause protein, like truncated protein, or the dysfunction. The protein will not function anymore, or they may cause the kind of nonsense immediate decay. That means the gene just will degrade at messenger RNA level, so that you don't have the messenger RNA or protein product. So that can cause very strong effect. They are highly penetrant. These are handful of genes here, but they are extremely rare. The risk variant those genes are extremely rare. So if you summarize those different genetic risk factors, this diagram can describe that very well. For example, here you can see the GWAS loci, the very common. The population frequency is usually more than 5%, or at least 1%. But the odds ratio is very low. It's like a little bit more than 1.0. Most of them, they're less than 0.2. So this is a schizophrenia 287 loci. Now, if you look at the rare variant side, you have extremely rare CNV here. The most common one is 22q deletion, also 2016p duplication, also confers schizophrenia susceptibility and other CNV regions, as you can see here. But they are very rare. And also some a rare protein coding variant that it can change protein function, or cause protein, the lack of protein product. So those risk variants also confer strong effect to schizophrenia risk. So you have two sides of the risk spectrum here. Now, the genetic discoveries during the last decade or more, a little bit more, really provide unprecedented opportunity for us to understand novel disease biology, to really look into how we can, translate the genetic discovery into novel treatment. So the first thing we need to do is to better understand the disease biology underlying those genetic associations. So that's what I have been doing during the last 10 years, called using functional genomics approach. Now, what's the functional genomics? So, as you know, this is the cell. Inside a cell is the basic, building blocks for our body, right? So inside the cell, you have a, genetic material, genetic makeup, which are double-strand DNA wrapped around those histone proteins. And then you have those genetic material can be transcribed into messenger RNA and then can be translated into protein. And those proteins kind of, confer the cellular phenotype that directly relate to, the clinical symptoms. If we talk about the genetic risk variant, basically at the genomic level, has to be translated into protein, which is protein level, and then connect to the phenotype. And in the between, you have transcriptome, basically the messenger RNA that is that will be translated into protein. So and also you have protein degradation process. You have product degraded from protein and other metabolite, so they call the metabolism. So combining this layer of information, a multi-omic, genome, transcriptome, proteome, metabolism, and also they interact with environmental risk factors here, they confer, susceptibility to the disease through a cellular phenotype and the, clinical symptoms. So the whole process basically, studying how genes and their product function to affect biological process and phenotypes can be considered conceptually as a functional genomics. So in order to understanding those disease biology, we need a model system, right? So we knew from years of research, animal model, let's say you can use animal model to model psychotic symptoms. You can use postmortem brain to understand how synapse pruning, reduce the dendrite or reduce the synaptic protein puncta in density in patient's brain. And of course, you can use a live brain for functional imaging, MR, functional MR to study brain connection. But these are models, each model has their pros and cons, as at least here. But today, my research focus has been, using stem cell derived model. So we use stem cell to differentiate into neurons and also brain organoid, you can call mini brain, and then to study, how the genetic risk factor affect the cellular phenotype that may be relevant to schizophrenia and other NPD. So I'm not sure if you are familiar with the stem cell, our iPSC induced pluripotent stem cell. So this is actually a Nobel Prize winning this kind of technique. So how you can derive stem cell from a somatic cell, for example, a drop of blood. So basically, you can take a drop of blood or fibroblast of, skin biopsy. You can, using biochemical or some, express some transfer factors, genes into the cell, and then, convert them back to pluripotent stage. We know, human body, let's say we all originate from a single fertilized egg, right? So that is pluripotent. You have embryonic stem cell there. So basically, the iPSC has the same property as ESC, embryonic stem cell. So from there, you can imagine from stem cell, you can redifferentiate them into, neurons, some other cell types. Of course, neurons would be the most relevant cell to schizophrenia and other neuropsychiatric disorders. And then, if we use that disease model, for example, you have a CNV carrier, you generate iPSC from that CNV carrier patient, and then you can differentiate neural model to study how a gene, which gene affected the cellular model that are relevant to the disease, and use that. You can, use that as a model to screening for screening drugs. So which cell type are relevant for schizophrenia? So this is from the GWAS data actually. If you look at the single cell RNA sequencing data here, so each dot here is a cell from the brain, postmortem brain. As you look at their expression profile, you cluster them into a UMAP, T-cell map here, and then you look at the cell type specific expression of each gene, and you look at enrichment, like we talk about GWAS association. You look at those GWAS association in the enrichment of those GWAS signal in each cell types. What you can find actually here is from the Nature 2022 PGC3 consortium paper. You can see here, neurons are most relevant cell type for schizophrenia because schizophrenia GWAS association signal are most enriched in those cell types. But this is not necessarily mean, other cell type are not important. For example, Dr. Marvin mentioned synapse pruning. The cell type important for synapse pruning actually is microglia. It's the only immune cell, residual immune cell in the brain, right? So microglia is responsible for pruning the synapse, of the neurons. So yeah, other cell type may also play important role, but the most genetically relevant cell type are neurons. So now we have a model. We have a stem cell. We can differentiate the stem cell into different type of neurons, glutamatergic neuron, dopaminergic neuron, or GABAergic neuron. And we can also differentiate those cells into a stem cell into microglia, as I mentioned, important for synapse pruning, and also astrocyte, which provide nutrient for neuron maturation in the brain. And usually, if you in the dish, the culture in the dish, this 2D culture, they are very pure, so that can easily know to study gene function use this kind of 2D model because they are very relatively pure. Of course, there's the limitation because in the brain you have a different type of cells they may interact. So that I will talk about the brain organelle that have multiple cell type that maybe better recapitulate the brain cell interaction. So with 2D model, we also can 2D and 3D iPSC model. We can also combine with the another technique, maybe called I'm not sure how many of you are familiar with this, called CRISPR gene editing. Basically, you can add the gene, add the SNP here, use CRISPR technique. So with the CRISPR technique, you can introduce a patient-specific mutation in the healthy control stem cell line, and then introduce to neurons or organoid to study the function of the gene. Or you can also use patient-specific, iPSC, and then correct risk allele to non-risk allele, so that you can compare, what changes are because of the risk factor, genetic risk factor. And in the lab, basically, we do kind of single-cell multi-omic profiling. We can do high-content imaging of those cells, differentiate from the stem cell, patient-specific or genetically modified, and then we can use the model to identify druggable gene pathway. If a valid model has been established, we can use them for high-throughput drug screening. That's the purpose, final goal of this iPSC modeling. So, with this iPSC-derived neuronal model, the first question we try to answer is, how to make sense of the GWAS associations, right? So we have so many, hundreds of loci. Which genes are really causal? How they cause the disease? To address that question, actually, there are quite a few number of challenges. At least some of the challenge here, as you can see, this is one of the, those hundreds of loci, right? You can see each dot represents a SNP marker. So, and each here is the gene track. You can see, the association signal is, similarly stronger for hundreds of markers, common variant, in the single locus. Now you have multiple genes here. And you remember, for example, if this is the causal SNP, it might regulate a gene, let's say 500 kb away. So you really don't know which gene and which are causal variant, which are risk gene, causal gene here. So that's the challenge. Another challenge is, most risk variant here, they are on non-coding part of the genome. So we talked about those highly penetrant protein coding risk variant. They alter the protein function. But most of the GWAS risk variant, they don't alter protein function. They regulate gene expression. That means they change the gene expression. Maybe, very mild change, like 20 to 30% gene expression change. So can we really, detect, the biological function with that kind of, for common variant of small population effect size? So those questions are challenging, comparing to study, those highly penetrant protein coding rare risk variant. So as first of the, this is one of the first study in the field. We try to use open chromatin as a concept to prioritize those GWAS association variant, disease-associated GWAS risk variant. Try to connect genotype to phenotype, use kind of open chromatin as a concept. So we mentioned that earlier, like our genome genetic makeup is double-strand DNA, wrapped around those histones. So you can see in some part of the genome they are more open. So in ATAC-seq kind of high throughput sequencing technology, you will see the sequencing rate here is show up as a peak that are called open chromatin region. So the hypothesis is that the genetic risk variant under those peak, they are more, they are in open region, they are more functional, they're likely to be functional. So use that kind of approach, we try to prioritize the GWAS risk variant among those 100 loci. So you can see here we can substantially narrow down the likely functional disease causal variant using this open chromatin approach. As a proof of concept, we also show one of the loci, MIR137 here, we use CRISPR edit the risk allele, and we observe they can change the gene expression of that gene by itself. MIR137 is a small RNA, and small RNA can also regulate many other target genes, including some glutamatergic receptor genes actually. So we showed it can regulate gene expression, MIR137 itself, and also some other target, and also can change the neuron morphology in terms of the dendrite complexity and also the synaptic protein maturity. So this is earlier study we use this open chromatin approach, try to connect the non-coding schizophrenia risk variant to cellular phenotype that may be relevant to disease. And later on, we kind of use a more systematic approach, try to identify, genome-wide, which of those GWAS risk variant they can affect the chromatin accessibility to regulatory factors. So the approach here, we use ATAC-seq. We call it allele-specific open chromatin. So basically, ATAC-seq, if you compare a heterozygous SNP, individual with a heterozygous for a GWAS risk variant, you have two alleles. Now, if you have sequencing rate of one allele is more than the other, more likely to be functional because one allele may be more accessible than the other allele may be functional. So with that concept, we use 20 iPSC line to generate iPSC, differentiate them into dopaminergic neuron, GABAergic neuron, and glutamatergic neuron, and then we profile the ATAC-seq open chromatin regions, and we called ASOC, allele-specific open chromatin variant, and we study the enrichment for GWAS association, and also we use kind of CRISPR and single-cell RNA-seq to edit those specific SNP to see whether the risk variant itself can really change the gene expression and neuronal phenotype. What we found was, there are a lot of ASOC, meaning risk variant, they show differential chromatin accessibility. and they are cell type specific, as you can see here. And this is one of the GWAS loci called VPS45 locus. It has the risk variant you can see in glutamatergic neurons show strongest ASOC, meaning one allele is more accessible than the other allele in glutamatergic neurons. So we used CRISPR editing to edit the SNP. We have so that we can have the iPSC line now heterozygous for risk allele, non-risk allele, and also the heterozygous, one risk allele, another is non-risk allele. And then we differentiate into neuron. We look at the gene expression, which gene affected by the SNP editing. What we found was, VPS45 itself, you can see this here is the VPS45, this is the SNP site. VPS45 was affected. Interestingly, actually, another gene kind of transcribed in the opposite direction is called long non-coding RNA. That means it does not encode the protein sequence. It's coding kind of a long non-coding RNA. It can regulate other genes expression. So also, its expression was also changed. And interestingly, there's another distal gene, but kind of 100 kb away, I think, also affected by the SNP editing expression. So we think, it's interesting because one single locus, you can have multiple genes that may be involved in regulating the gene expression that may be relevant to the disease. So we test the hypothesis whether those risk genes, the 3 risk genes here in the single locus, have synergistic effect at the transcriptomic level. What we did was we kind of knocked down, each gene individually, and then we did another combinatory triple knockdown. So we compare, if you based on the additive model of genetic risk, you can calculate, 1 plus 2 plus 3 equals, 6. Now, if you have combinatorial triple knockdown, you would expect you have more upregulate or more downregulate. It's not like equal to the additive prediction here. So we can see here there are 1,000 genes more upregulate or downregulate here. If you look at the gene pathway, involved in those more up or more down-regulated, you can see they are enriched for schizophrenia-relevant ontology terms, like schizophrenia GWAS, MIR137 pathway, bipolar differential gene expression, and the same for the more down-regulated gene here. You can see including antipsychotic drug treatment. So that means, this is kind of interesting. One single GWAS locus, you have multiple genes involved. They can show synergistic effect that contribute to disease-relevant, phenotypes. We also studied the neuronal morphology chains, which I didn't show you here. You can read the paper if you are interested in. So what I'm talking about until now is about common GWAS risk variants. So how we can use iPSC to model, those common GWAS risk variants. It's challenging because, they have small population effect. Actually, in the field, more low-hanging fruit, or more people would like to do is to model highly penetrant, protein coding risk variant or CNV, because they are highly penetrant. From cellular phenotype, if there's any phenotype change, it's easier to interpret. So this is the summary. Basically, your iPSC model, you can model some. Mostly related to neurodevelopmental disorder like autism spectrum. But for schizophrenia bipolar, you can also use them to model, some specific, genes, genetic defect. So in our lab, we also did some exploration on this part. Basically, we have been focused on a 15q 13.3 deletion that associated with schizophrenia. So what we did was, we tried to, because as I mentioned earlier, each CNV region has many genes, they span multiple genes. You don't know which is the driver for the phenotype change, right? So in order to identify that, we did a kind of deep sequencing. So here, basically, you can see we did deep sequencing in those CNV regions systematically. At this region, we found some nonsense mutation, which, cause stop codon that can cause protein truncation or cause non-sensitive decay of the gene. And then, those mutations we found only present in schizophrenia cases. Very small number, but it only present in schizophrenia cases. We hypothesized that, this gene may be the driver gene. Those nonsense mutation may cause protein, loss of function of that gene may contribute the cellular phenotype that may be relevant to disease. So we kind of use the similar approach I talked earlier, use CRISPR editing to introduce this patient-specific mutation into a healthy control iPSC line. And we generate, the iPSC and then differentiate to glutamatergic, which exciter neurons, and we study their neuronal phenotypes, and also study the gene expression, transcriptomic changes. This is one of the phenotype we assay. You can see here we look at the neuron firing in terms of the neuron, how they are more active or less active. As you can see, for that deletion, the mutant allele caused the neuron firing, substantially reduced neuron firing in glutamatergic neurons. So that means, this is likely to be kind of the risk allele likely to be contribute to, reduce the neuronal activity, maybe consistent with schizophrenia. So this is the one example how we use iPSC to study a rare variant. We also, like I earlier mentioned, we also use 3D. Why we use 3D model, which is also called the mini brain or brain organoid? Because they have multiple cell types. The good thing is use 2D, they are homogeneous, you can get very pure cell culture, neuron culture, but in reality, the model that can be better recapitulate the brain complexity is, the organoid, also called the so-called mini brain. So how to do that? Basically, you have iPSC, you grew them in a dish, you keep culture, you use shaker or not shaking, you can let them kind of grow in a group, and at some point they form into like a ball-like structure. If you cut them, you stain them, they look like, they have some kind of, cortical-like structure. You have ventricular zone, you have deep layer, and, upper layer neurons. This is ventricular zone. So it's kind of resemble the brain, kind of a cortex structure. So with that, model, we kind of study a highly penetrant protein coding called truncating risk variant I mentioned earlier in the gene called SETD1A. SETD1A is a gene actually modify the histone methylation. So these are the risk variants identified in this gene associated with schizophrenia on the upper panel here. And lower you can see those risk variants that can also cause other neurodevelopmental phenotypes. The most interesting mutation, I think, is exon 16 because it's recurrent, as you can see. Some of them are de novo, but this one is like recurrent here, you can see. And also, enriched in exon 16. Now, so we use CRISPR editing, try to, introduce this risk allele into those healthy control iPSC lines. Here you can see we differentiate to, we have the CRISPR engineered iPSC line that carry the risk allele and non-risk allele, and then we differentiate to, cortical spheroid or organoid. And then we did transcriptomic analysis and also morphometric analysis, and also we measure the calcium imaging, which measure neuron activity. This is the confirmation of the, how the risk allele can cause reduction of the protein expression of SETD1A. You can see in mutant allele has 50%. This is heterozygous, or what we generate here is heterozygous. So you have reduction of the protein level. And then you look at the function, at the cellular level, you have reduced as SOX2 positive cells, and you have premature kind of a neuron appear in the early stage of organoid. So this kind of precursors neurodevelopmental effect associated with this risk allele. Now, a later stage, you can see more like cortical structure like morphology here. And then if you look at the calcium imaging, which is measured neuronal activity, this is individual neuron calcium imaging. And then you compare that case wild type, the mutant line, you see mutant line, mutant allele associated with reduced firing frequency in exciter neuron. Now this is a video shows you how, basically it shows you how the neuron firing. You can see, they are flashing, each cell in the organoid. So how we can use this model? So we didn't, reach this step yet, but this is example show you how you can use this model to do drug screening or to test, to validate the drug effect. So here you can see they used group from Stanford as a friend. He used kind of organoid cell, the cell dissociated from organoid to measure calcium imaging. You can see this is 20q deletion syndrome carrier. They show reduced calcium spike, calcium signaling capacity. Now, if you use antipsychotic drug, you can see they can rescue the phenotype to the control level. And if you stain those cells, you see they have quite abundant expression of dopamine D2 receptor, which makes sense because they are the target of antipsychotic drug tested here. So with this kind of model, you can imagine we can use that to screen novel drugs, novel chemicals that may be more effective than classical antipsychotic drugs. So up to now, I have been talking about how we can use iPS-derived model to use CRISPR editing to edit one gene at a time to study how a gene affects the function, cellular function. Now, actually, in NIH, we have a consortium called SciGene, try to kind of scale up the assay, try to assay the neurodevelopmental and neuropsychiatric disorder risk gene in large scale. We, as part of the consortium, Dr. Jiping Pong and Rutgers and I, we call that the assay center called MIND. We named it MIND. So we propose to kind of create loss of function allele iPSC lines for about 200 genes in 6 different donor genetic background, and then we differentiate into neuron to study their function, like electrophysiology and transcriptomic properties. So up to, this is as of end of last year, we have 45 genes on 6 donor line generated, but actually now we have close to 100 genes generated. So this is the paper published Stem Cell Report last year. We kind of summarized what have we done at that time. So basically the pipeline was, we use CRISPR editing to introduce premature stop codon, which is kind of model the, the ultra protein truncating mutation in those neurodevelopmental risk genes. And then we kind of bank those iPSC line, engineered iPSC line, and we differentiate into neurons, different type of neurons, exciter inhibitor, and co-culture them with S-glia cells. And we do high content imaging to look at whether they can change the neuron dendrite or synaptic function. And we also do single cell RNA-seq, try to identify what's the convergent gene pathway systematically across those hundreds of neurodevelopmental risk genes. Just to show you some of the results from the high content imaging here, as you can see, SHANK3, you heard earlier from Dr. Cook, is important for autism and cause reduced synaptic function. And here, indeed, we kind of validate that, use this kind of high scaled approach. And we also, and this is one of the novel schizophrenia associated genes. As you can see, we found reduced synaptic dendrite synaptic function, and also synaptic puncta density, and also the reduced neurite outgrowth, which is kind of consistent with the reduced neuron function in schizophrenia. So to summarize, basically, during the last 10 to 15 years, through GWAS, whole exome sequencing or whole genome sequencing, we have identified many, thousand common genetic risk variant, but they have small effect size associated with schizophrenia and other neuropsychiatric disorders. Another side of spectrum, we have a rare risk variant in protein coding region that it can alter protein function or, cause loss of function, basically. but they have large effect size. So NPD, represented by schizophrenia, I would say is most heritable among those NPD. They are polygenic, meaning they have many risk genes, hundreds of thousands risk genes, each contribute a small effect. Individually, they may not, matter, but combining them, in pathway level, they may, they may contribute to substantial to, disease stability. Also, those genetic makeup are shared across major psychiatric disorders, as I showed you earlier. And, the common risk variant, as I showed you earlier, is mostly non-coding. Various, small percentage, they may change protein function. They likely to affect the gene expression and chromatin accessibility. That means they are regulatory variant. A functional genomics approach, using iPSC in combination of the CRISPR editing, as I showcase here, they may help to connect the phenotype versus to genotype, connect the genotype to phenotype. It will help us understanding the disease biology underlying those GWAS associations and also the rare association with the rare risk variant. Lastly, a scalable assay for those neurodevelopmental and neuropsychiatric risk gene underway, which I think they can help identify some convergent, or divergent gene pathways that may be, shared by different psychiatric disorders, and maybe also specific to some, one specific disorder. Those information, obviously, they can be exploited to develop more tailored clinical treatment. With that, I thank you for your attention. These are the people, really working in the lab. Thank you very much.
Wow, thank you. Questions? Rob?
Could you predict future state of CRISPR gene therapy for psychiatric disorders?
So I'm just going to repeat the question and then ask Jubao to come up to the microphone. So the question was about being able to predict CRISPR and how it might impact psychiatric disorders.
I think, that's a fantastic question, I think. Even for, Alzheimer's, I also study Alzheimer's disease, use this iPS model CRISPR editing. Even for ApoE4 now, people have been talking about to use, use CRISPR editing to correct gene ApoE4 to ApoE3, ApoE, That's, ApoE4 has a very strong, penetrance, basically, kind of more familiar than schizophrenia. So I think it would be challenging for schizophrenia and neuropsychiatric disorders. But for those rare variant, I think, extremely rare variant, definitely is doable. And it's, I think, in terms of personalized medicine, I think it's worth to do. I think it's very promising in the future, I think. But it would be very challenging for common, like a risk variant. Yeah.
Super. So I'll ask one last quick question, sort of follow up to that. If you could comment on the overlap between neurodevelopmental disorders, the neuropsychiatric disorders, and neurocognitive in terms of sort of thinking of Alzheimer's and things like that, where you see some of these genetic linkages, and are there any common threads between those three clusters of disorders?
Actually, I think, I'm not expert studying, cross disorder, like, there's a consortium under like PGC, they specifically analyze this cross disorder genetic overlap. But if from the analysis, you do see substantial overlap, across those different orders, different disorders and the spectrum, symptom spectrum. Yes, I think the simple answer is yes, I think. Yeah, super.
All right. Thank you.